Good afternoon. This is Qian. Today I'm going to present our latest work on probability modeling of single-cell RNA velocity. First, I'll introduce some biological background about our study. Cell phase is the core concept for understanding cell development and cell reprogramming and cancer cell progression. And the typical biological question can be included on the following three. How does one cell transform one cell state to another cell state? And what is the driver gene of such a state transition? The third, can we predict which of the cells would be the future cell state? So given this question, usually people will profile single-cell RNA data to study them. So how shall we predict the cell phase from single-cell RNA data? There are four categories of methods developed for this open question. The first is about trajectory inference method. This method captures the continual variation of cell state using tree or curve structure. The second method is called cycle tree. Cycle tree uses the expression number per cell to predict the differentiation level from a given single cell data set. The third one is called single cell lineage tracing data set. This method combines the lineage barcode information together with the transcriptional state in the embedding space to track the cell lineage and the state across physical time. The last one is the object we are focusing today, is called Probabilistic, which uses splicing and unsplicing information to predict the future cell state of the current cell state. So given that, what is R velocity? So R velocity is based on the prior knowledge of R processing and molecular biology. Adding such a figure, it assumes each gene's R processing is an independent reaction process, and each reaction's transcriptional splice and degradation form a sequential kinetic process, which is encoded by the alpha, beta, and gamma parameter. Now let's look at the input of the R velocity model. We can decompose the RNA rate count into the splicing rate count and unsplicing rate count. And given such input and the model, we can try to infer what will be the transcriptional rate along the time. And based on the inference of the kinetic pointer, we can predict the gene expression of splicing and unsplicing along the time. We can also visualize the splicing and unsplicing in a two-dimensional space. And we visualize in a curve. The curve is called phase portrait curve. The R velocity can be further interpreted as the distance between the prediction and your observation. So here we show two successful applications of R velocity in the part publication. On the left, it is benchmarking data set from a snapshot of single cell RNA data of the neuron genetic. So the author identified multiple lineage from the neural genetic process. So it can start from the neuroblast, which is the stem cell, toward multiple mature terminal cell phase. On the right, the author leverages time series design with a one-shot labeling assay NTC to study the in vitro hematopoietic assay process with a chemical labeled RNA molecule using the R velocity model. Even though this model has been successfully applied in different biological concepts, but there are some remaining problems. The first challenge is about the complex kinetic with multiple trajectory problem, and each of the kinetic can form one trajectory for the biological process. And the current R velocity model, because of its limitation for modeling only one trajectory each time, it may predict the reverse order as on the right figure. The second challenge is a sample with a fully mature blood cell type, and the current R velocity model designed for modeling the dynamics from the single cell RNA data. But such a static population can be mistakenly characterized as a dynamic. And the third problem is about pre-processing. As we will show later, the pre-processing choice would be dramatically change the direction you have predicted. So what shall we do? We need a certain animation for all the prediction of the vector field. And this can denote how much we trust for each of the direction. Then we can thresholding for the confident vector field. So our solution is called Pyro-Velocity. It is an end-to-end Bayesian model. So we can look at our solution side by side with the previous publication. So the input is the same. It's still splicing, unsplicing, raw rate count table. And the upper diagonal show the workflow of the previous R-velocity model. So here is the problem. The whole workflow design is ad hoc. It constitutes with data smoothing and velocity estimation per gene independently, and with over 10 different steps of processing. What shall we do? So in the bottom part, we show the workflow and our innovation. So our model is a generative model that can directly input the raw rate count without any smoothing and ad hoc transformation. The second that instead of modeling the latent time for each gene independently, we can model the synchronized share time across all the genes together. And this much simplifies the workflow. We don't need any of the post-processing in the downstream analysis. The third one that our framework provides several diagnostic plots to assess the dynamic and its uncertainty for the sample you have sequenced. So briefly, our framework provides 4 major outputs. The first 2 outputs are from the vector field and the share time together with the uncertainty. The second one is that our generative model can predict the correct development order of the share time. And the last one that we can identify the key gene associated with the cell fate determination, and we can show the rainbow plot to show its correlation with the share time. Next, we try to use several benchmarking data sets to study our framework. So the first benchmarking data set is from pancreas development single-cell RNA-seq data. This sample is constituted with 8 different cell types, and they have well-known development order, which starts from the ductal cell toward the pre-inducting cell and the inducting cell, which including the beta and the alpha cell. And the current R-velocity model, such as SVelo, had output the point animation of the vector field that recover the correct cell fate. And at the same time, the current velocity model can prioritize some of the important markers in the inducting cell, such as the CP gene and the insulin gene, insulin 2 gene, and also some important marker in the ductal cell, like in the ADK. So, however, if we try to zoom in the vector field for a small proportion of the cell types, such as epsilon cell, the direction actually largely impacted by the typical pre-processing parameter, such as how many neighborhood cells we selected for the KN smoothing, how many of the top variable genes we have selected for the analysis, and the extreme case can even reverse order for the future cell state of epsilon cell type. Then what shall we do? Well, what we need to do is apply our potential solution, Pyro-Velocity, to such a benchmarking data set. First, if you look at the left figure, it's a qualitative comparison of the stream plot, which is representation of the vector field for R-velocity downstream trajectory inference. So most of the cell population look very similar across the previous R-velocity model called SVelo and our solution, except for a very small area in the ductal cell. Then we try to look at the typical example of the individual cell vector field from 30 potential samples from our generative model. We can see some of the area are quite certain. They point to almost the same direction. For some of the area, they are quite random. They point to almost every direction. So how could we design a metric to study uncertainty? So we try to develop 2 metrics based on the circular statistic. The first circular statistic is called circular standard deviation. So 0 means it's quite confident, almost point in one direction. 360 means it's quite stochastic, can point to any direction. As you can see from the figure, the left one shows the individual cell level of the circular standard deviation. The right one shows grid cell level of aggregation of the vector field. That the most uncertain area highlight the region of the ductal cell, which is constituted with the cell cycle, and from the bifurcation cell that within the inducting cell type. The other metric is called Rayleigh test. It's a test whether a certain cell can have a unidirectional trajectory in the future. And the result based on the Rayleigh test is very similar, have a large overlap with the circular standard deviation. So the next metric our model can output about share time. So if you look at the heat map with the UMAP embedding, they have a very similar gradient across the different cell types. So we try to quantify our metric based on the Spearman correlation with a silver standard called cytotrace. It's kind of state-of-art method published on the science. So our velocity model is slightly better than what is published on ISMB. And again, we can also look at the standard deviation of the share time, which represents the uncertainty of such a scalar value. We can also use such a standard deviation to highlight the most uncertain area would enrich in the cell cycle stage. So the next one, given the share time, we could try to prioritize the cell phase marker based on the correlation of the splicing gene expression level with the share time. And also based on the goodness of fate on our velocity model. Here in each of the dots in the scatter plot represent one gene, and we can highlight the most positive correlated gene in such a plot. We can even visualize the fate portrait based on the denoised data of our model. You can look at that they have a clearly separated different cell type in the fate portrait space. And we try to create a new visualization, which is called rainbow plot. We order the cell based on the x-axis, which is the share time. The y-axis is still the gene expression from the splicing. And again, we can clearly say there is strong pattern of gene expression increasing along the cell type, and the different cell type can be clearly separated after ordering by the share time. So our model has working pretty well on such a positive case. We want to test whether it can work on a true negative case, which is a data set we have introduced in the introduction session. So this is the same study population for high multiple axis. We want to use that benchmarking whether our model can have a statistical diagnosis of the data. So apart from if you look at the posterior mean of the trajectory, we can see they form such an artifact of linear trajectory starting from the CD19 positive cell toward the other cell type. But upon that, our model can provide a statistical test for the circular statistics. If you look at the individual cell, some typical individual cells, they almost form a random direction trajectory. And if you look at the circular standard deviation and the statistics test from the Rayleigh test, almost all the cells stay at the static state. So our model can conclude this is a steady population of the fully mature cell type in PBMC. So the next we try to move to an even higher bar, the most challenging case from the single cell lineage tracing experiment. So the left figure shows the experimental design from the Larry lineage tracing experiment. We have 3 different time points. So the cell had been initially barcoded with the lentiviral barcoding. Then we harvest the cell for single cell sequencing. If you look at the left figure, which is the embedding contracting from the splicing expression level, we can clearly see different clone lineage on the embedding space. So next, we just want to use our framework and also the clone barcoding derived progression to do a qualitative comparison across different vector fields. So the leftmost vector field is the one which is derived from the transition matrix of the clone barcoding. The middle one is the vector field from the SVLO, and the right one is our result from Pyro-Velocity. We found that there is actually no perfect model. Sometimes that our model and SVLO can do a much better job than the clone barcoding for the neutral field. And sometimes for the basophil cell type, that our model cannot have a good signal for predicting the right trajectory. And sometimes for the megakaryocytes, that our model and the clone barcoding model can do pretty decent job, but SVLO cannot do that. So given vector field do not have a gold standard representation, we try to benchmarking our algorithm with a fate potency score, which is developed on COSPAR. This score can be regarded as a gold standard to benchmarking the latent time or share time. Based on such a metric, we can say that our method have a positive correlation of about 0.65 with a fate potency score. But for the published SVLO method, they even have a reverse order, like a negative correlation. So that since there is no gold standard for multiple trajectory, so our future direction will focus on the complex trajectory with several statistical diagnostics. And the action actor can be divided into 3 components. The first one, we need to develop an end-to-end fully generated model for developmental trajectory and the self-weight decision from the raw data. The second, we need to incorporate the intrinsically stochastic dynamic model to account for the bifurcation and multiplication event. The last one, given we have multiple data sets from different conditions, we can have an estimation for the differential self-weight. So even though our model is still far from perfect, we still have some contribution that worth mentioning the take-home message. Our Pyro-Velocity model provides the first end-to-end solution to model the RNA velocity and the self-weight from the raw weight count and synchronize the estimate from all the gene. And our model provides a natural estimation for the uncertainty level for the recovery share time and vector field, together with several diagnostic plots. The last one, we have an automatic way to recover and prioritize the important genes that can interpret the self-weight and dynamics. So the last but not least, and I would like to thank all my mentor, Luca Pinello, and Professor Dave Langelaw. Thanks for their patience and the support for all the projects we have made recently for the past 3 years. And also I need to thank Eli Bingham, who is the inventor for the probability modeling. It helped us a lot for resolving such a complicated model. And also we need to thank the inventor of Parallelity, Joella. He gave us some positive feedback on the model. And the last, I need to thank about my wife and my mother. They take the lead to take care of the baby. Then I have the time to explore the model. Yeah, thank you.
Wonderful. We have time for one question.
Yes. Yeah, I think that's a good question. So as I mentioned in the introduction, our velocity model, in theory, it can be designed to be model one trajectory. So the reason why we say there's the uncertainty in the bifurcation point, because all the endocrine cells form a linear terminal self-weight. So essentially, if you look at all the other data like neurogenesis, like hematopoiesis, you use a one trajectory model to model the multiple lineage. So the intersection region or the multiplication point would always be very uncertain because we didn't incorporate the stochastic in the model. So there is that uncertainty caused by the biology, also caused by the model predicted error. Yeah. Yeah, thank you. Yeah.